Hi everyone, and I welcome you all to this very special session on antimicrobial resistance. This is obviously national news because it has been addressed by our very own Prime Minister. That is why today we have held a very special session, short session, just to update and jog your memories about antimicrobial resistance and what it is about. Of course, you have read all about it with me in the classes in your MBBS years, and also if you're preparing or a practicing doctor right now, you must be very aware about this pandemic like situation which is happening in the healthcare world. If you have joined me, please come and say hi in the chat box. Hi Munir, how are you doing? I'm very happy to see all of you. Really excited for this session. So basically what has happened recently is that the PM has addressed the nation saying that antimicrobial resistance is an all-time high. And why has it become such an important problem that the leaders have to address it? Even recently in a journal which is called as the Lancet, which is one of the most prestigious academic journals of the medical world. Hi Manisha. They have said that across a survey across India, they have realized that 80% of the Indian patients carry multi-drug resistant organisms. You must remember that we refer to them as MDROs, and you see this word written everywhere. MDROs, multi-drug resistant organisms. These organisms, these bacteria have found antimicrobial is. Hi Manisha, I'm good. How are you? Hi Deepak. Very nice to see all of you. Of course, very good to see all of you here. Right. So these MDROs, multiple drug resistant organisms, many drugs are not able to work on them. And this is a common scenario every day when I work in the labs, when I work. Okay, Manisha, great, good job. Keep it up. Hope to see you very soon in MBBS. Listen very carefully to this because this session is very important for everybody who's watching, medicos or non-medicos, and also great opportunity for the medical students to revise. Every day, on a daily basis in my laboratory when I'm sitting and when I'm discussing with clinicians treating the patients, every day we come across patients who are in critical care in ICUs. Even in the OPD basis, patients come to us, their samples come to us, and we find that the infections that they have are from microorganisms which are resistant to a whole spectrum of antibiotics. When we test in the laboratory, we test against almost 8 to 9 different classes of antibiotics, and none of them seem to work against these patients. And a study like Lancet claiming 80% of the Indian population, that means almost all of us are living around with these multi-drug resistant organisms in our body, outside our body, in our flora, in our gut, in our patients, everywhere. That is the most dangerous situation you can imagine, because in the last 30-40 years, no new antibiotics have really come up because research is not happening on that. And the pace at which antimicrobial resistance is increasing in the bacteria, we have no treatment options left. So let's quickly talk about what this antimicrobial resistance is. Let's understand what it is and what can we do to help this situation. Of course, there's a lot of awareness going on, and they have talked about that there are ways in which we can protect ourselves, which we can protect our families. The first and foremost thing that we have to take care is whenever we pop antibiotics like candied pills, we should stop doing that. I mean, this is a common thing that we hear in our houses, that our family members and everyone says that, oh, my fever doesn't go away if I don't take antibiotics. Quacks and many healthcare practitioners, which might be registered or unregistered otherwise, they have this habit of prescribing antibiotics left, right and center. You feel the tiniest pain in your throat, you feel the slightest of sore throat, you start popping azithromycin. Azithral is a very common antibiotic which everyone is taking. Then over the counter prescriptions, in our country especially, you don't need prescriptions from doctors for many high and broad spectrum antibiotics. You must all have heard of the pill Augmentin. It comes with many brand names such as amoxiclav is the salt name. Okay, you must have heard, it must be lying around in your homes also. Every time you get the slightest of fever, you pop a pill of Augmentin. Every time you feel the slightest throat pain, you pop a pill of Augmentin. You're feeling crummy along with paracetamol, you just take Augmentin. Do you realize augmentin is a broad spectrum antibiotic? There was a time not too far away, not too much in the past, when we used to give augmentin as a surgical protection in patients. When we would take patients inside the OTs, even today we do that, to protect them in a heavily sterile environment. When we are cutting open a patient, then to protect the patient against microbes, we give augmentin. Such a broad spectrum antibiotic we are taking in the form of tablets every now and then. And we don't care about the fact that it needs to be taken 3 times a day for 14 days. We take 1 or 2 tablets, then we feel fine, we forget about it, we move on with our lives. Because common misconception is that now my symptoms are fine, why should I keep taking the antibiotic? That is the biggest problem right now that we are facing, that the biggest contributor to this antimicrobial resistance and emergence of these MDROs is this unauthorized, unjustified, unsupervised popping of antibiotics by common people without prescription of the doctors. There are also other reasons why these MDROs are spreading, and we will just come to them. How do these MDROs even come into play? How is me taking an antibiotic affecting the population and creating 80% population to harbor these MDROs just because I am taking one augmentin antibiotic? We will talk about that too. So basically, whenever we are over prescribing the antimicrobials, like I said, there is selective pressure by the antibiotic on the bacteria. What happens? You must have heard this saying in English, what doesn't kill me makes me stronger, right? So let's see this. This is a red colored antibiotic, and I have given this to this mixture of bacteria which is there. There are some red and blue bacteria, and you can see that the red ones are sensitive to this antibiotic, so they are dying. But the purple ones are just a little bit stronger. So what have I done? I have taken the antibiotic. I have not given it for the complete dose. I have taken 1 or 2 tablets and I have moved on with my life because my symptoms got better, which would have gotten better on their own anyways. But I did not have time to wait. I am too crummy. I pop one augmentin and I move ahead with my life. What happened? That augmentin went inside my body. It killed some bacteria, but some strong ones who needed a stronger dose, who needed the proper dose which is as prescribed by doctors, they saw the antibiotic. They felt the pressure of it, but it was not enough to kill them. So they are now in the selective pressure of this antibiotic. Now they will become smarter. If I send out to you in the jungle and you spot a tiger, the tiger has not killed you, but you have spotted it. Now you will become smarter. You will find ways to protect yourself. You will find ways to evade the tiger. Similarly, these microbes, these bacteria are extremely intelligent beings. That is the reason that the humans are very recent in the evolution, just few thousand years old. But these microorganisms have survived millions of years of this evolution on this earth because they're very fastly adapting and they're very, very intelligent. So when you put that selective pressure, you see how this purple bacteria has realized that this antibiotic is roaming about in the body and I need to protect myself. So it will devise some ways. Very, very fast they mutate their genes and they attain the capacity, the ability to resist the effect of these antimicrobials. So it has now become a superbug. The superbug has a shield and it is protecting the antibiotic. Again, the next time I get the infection, probably the same thing happens. Continuously, I am doing this for many years. I am just popping one pill every time I get a slight bit of fever, slight bit of sore throat. What happens after 10 to 15 years? This purple bacteria has become extremely strong inside my body now. Now it will cause me some infection which will just not budge. Right? That infection is just not budging now. You pop more augmentin. You pop it for 14 days. You will now take another higher order antibiotic. Nothing seems to work. Why? Because now in this war, the bacteria is winning against the antibiotic. This bacteria now will transfer the same genes to another bacteria and they will produce a whole family of superbugs. And before you know it, your body is completely infested with these superbugs. And that is what is happening, what Lancet is saying, that 80% of Indians are harboring these superbugs inside their body. because of this left, right, and center overuse and overprescription of antimicrobials or antibiotics. Taking them when there is misdiagnosis of viral infection. You have a viral infection, the seasonal flu, the flu we get with the changing season happens because of viruses, and we pop antibiotics. Antibiotics are antibacterial drugs. They act against bacteria. So if you will take an antibiotic in a viral infection, you're only killing the good bacteria in your body, and you are putting selective pressure on the bad guys, and they are going to become worse. Right? Patients not finishing treatment. Like I said, it was prescribed for 14 days. Then there are people who say, I don't like to take many medicines. So what will I do? I will take the medicine for 2 days and then I will not take the medicine. That is a very wrong practice that we do. Now, overuse in the livestock and fish farms and the cattle and the dairy that we consume, this is also a big problem. Even though this was supposed to increase production and increase the betterment of the world with the globalization, but all of these antibiotics which are being added into the animals with which then we consume, they also contribute to the selective pressure on the microbes. Poor infection control practices in the clinics. In the COVID time, you have all been aware of the masks and the PPE and the gloves and the quarantine and the social distancing and all the protective measures. Whenever you go to an ICU inside a hospital, they always tell you to remove your shoes outside, wear the apron. Sometimes we don't want to listen to them because we think, what am I? I am a clean person. It's not about being clean. These microorganisms are tiny. They're microscopic. You will not be able to see them. My hand looks clean, but there are trillions of bacteria on this hand even right now. So these infection control practices is very, very important. Inside the ICUs, inside the hospitals, there are patients whose immunity is weaker. You have come from outside, you're harboring lots of bacteria which are not affecting you because your immunity is strong right now. But you can transfer them to these patients. And then it becomes even more difficult to treat these patients. So all of these are the common scenarios which are really increasing the burden of antimicrobial resistance. Now, how do these bacteria take up this resistance? So there are two ways. One is intrinsic antimicrobial resistance. Intrinsic means by birth. So there are many classes of bacteria, different types of bacteria, just like there are many types of people, good people, bad people, short people, tall people, like that. Like that, there are microbes. There are two categories, you must all remember, the gram-negative and the gram-positive bacteria. They have particular structures, they have particular shapes, their cell walls have particular proteins and lipids and everything, which changes as the class of the bacteria changes. Now, some bacteria, by the virtue of birth, by the virtue of their structure, they have the ability to resist certain antibiotics. For example, vancomycin and gram-negative bacteria. You know gram-negative bacilli is the GNBs, your common Pseudomonas E. coli, this and that. Vancomycin is a high-order drug, which is a very common practice to start vancomycin in most of the patients which are admitted in the hospitals. Because you want to give some gram-positive coverage, when the patient walks into you, you don't know whether he has a gram-positive infection or a gram-negative infection, right? So you give vancomycin. But once the lab report comes and it tells you that a gram-negative infection is there, you need to stop vancomycin. Because gram-negative bacteria have an outer membrane in their cell wall. And that outer membrane does not allow vancomycin to enter inside the bacteria. So the antibiotic by nature, by birth, is ineffective for GNBs. That is intrinsic resistance. It is intrinsic, it is natural, it is by birth that the bacteria will resist this particular antibiotic. So as a doctor, as a clinician, it is your responsibility to find out which microorganism is affecting my patient and then give the appropriate antibiotic which is going to act on that bacteria. Then is acquired resistance. Acquired means when you move about. So when you are born, you have the intrinsic ability to feed, to eat, to drink milk, to eat. But you don't have the intrinsic ability to speak in a particular language. As you grow up in a household and they are speaking Hindi or English in front of you, you acquire that, you learn that. Similarly, when the bacteria are exposed to different types of antibiotics in their environment, they acquire the ability to resist them, just like we had seen in this Superbugs family a while back. Under the selective pressure of one particular type of antibiotic, the purple bacteria learned to resist it and became more and more resistant. That is acquired resistance, right? So this acquired resistance can happen because of a mutation, which very commonly happens with the TB bacteria. So this is a question. What type of a drug resistance is seen in Mycobacterium tuberculosis? Mutational drug resistance. Basically, there is one bacterium of TB, some mutation happens in the genes and it changes its nature and it becomes resistant to one particular type of antibiotic. How do we meet that? You must all know that we give the multi-drug regimen for treatment of TB. A patient of TB does not take one antibiotic. He takes a combination. HRZD you must have heard of. 4 to 5 antibiotics in a single combination are given to a patient of tuberculosis. Why? Because if in a moment the bacteria develops some mutation and becomes resistant to one type of antibiotic, you have the other 3 or 4 classes covering the patient. Because it is very important to stop TB, especially in a country like India where the burden is high, and also this antimicrobial resistance we are dealing with in a deadly disease like TB also. There is the XDR TB strains, extensively drug resistant tuberculosis strains, wherein there are very few options left and the treatment also gets prolonged up to 1 year, 1.5 years when we are trying to treat these patients. So it is that real and it is that dangerous. Then gene transfer happens. One genetic transfer is the simple one. I have acquired some mutation and through the gene transfer, I will pass it on to my child through the vertical transmission by birth. Same happens in bacteria. One bacteria divides into 2, gives its own genes into the 2 daughter cells that it has produced. That is vertical transmission, parent to child. What bacteria have this special capacity is called as horizontal transfer. That is the transfer between unrelated bacteria. This we had read in genetic transformation. Remember the gene transfer between bacteria, we had read about it. So bacteria have this special ability to transfer the genetic information in unrelated to bacterium just sitting next to other. Just like I am doing with you right now. Genes are nothing but codes. They are codons, they are information in the form of codes, right? I am speaking to you right now and my knowledge I am transferring to you via this medium, and this is happening horizontally. We are not related by birth. We don't have a parent-child relationship. We are unrelated. We come from 2 different lineages of parents. But as I am talking, information I am transferring to you. This is horizontal transfer. Similarly, this genetic information of drug resistance is transferred between one bacteria to another in the form of horizontal transfer. We have all read about conjugation, transformation and transduction. I also have a video on the same wherein I explain how these bacteria are able to communicate this genetic information with each other with small mobile genetic elements and also with the help of plasmids. And there are certain viruses which help them also. So you can check out those lectures. Of course, if you have read it, you remember conjugation, transformation, transduction are the methods of genetic transfer between bacteria, and that is very important for rendering acquired drug resistance amongst the bacteria. That is why once a patient is infected with a MDRO, we find that first you tested the organism, it was resistant to 3 or 4 classes of antibiotics. After some time from the same patient, you will take the sample, you will realize the same set of bacteria has now become resistant to 10 to 12 classes of antibiotics. Why? Because they have been able to transfer the drug resistance between each other and become more and more drug resistant as time passes, right? Now, how is this drug resistance acquired by the bacteria? What do they do actually to attain this drug resistance? So, the first and very important is the decreased uptake. Mechanism is decreased uptake. You all know that on the surface of the bacteria, there are the pore-in channels. These are the little pores which only allow some ions to move out and some ions to move in, right? So, the bacteria will become smart. They will change the nature of these pore-in channels in such a manner that this antibiotic will not be able to enter inside the cell only. Bacteria is a cell, right? So the antibiotic will not enter inside the bacteria only. Now what will the antibiotic do? Resistance achieved. Second very important mechanism is efflux pump. So efflux pumps are like vomiting. Antibiotic somehow achieves getting inside. Bacteria realizes antibiotic has entered inside me, vomits it out. Efflux pump sends it right out. Very, very important mechanism of efflux pumps, shown by many gram negatives such as Pseudomonas. Right? Second important mechanism, efflux pumps. Third, inactivating enzymes. Now it's war time. Bacteria will start producing enzymes. It will start secreting enzymes. Example, beta-lactamases. You must have all heard beta-lactamases. What are these beta-lactamases? Ases means to break down. These are enzymes which directly break down beta-lactam rings. Where is the beta-lactam rings found? Inside many important antibiotics. Example, amoxicillin. Amoxicillin is a beta-lactam antibiotic. You give amoxicillin, bacteria releases beta-lactamase. Beta-lactamase goes, breaks down the antibiotic only. Antibiotic destroyed, bacteria wins. Then you have target site alterations, number 4, very, very important. You all must have heard of penicillin. Penicillin was a wonder drug when it came about. In World War II, it saved many soldiers. Today, we don't give penicillin to anybody because there is hardly any bacteria left which is susceptible to penicillin. Almost all bacteria existing today in the infectious world has become resistant to penicillin. Something which saved lives not even 70 years ago, totally and completely rendered useless these days because bacteria has become so smart. What do? Of course, you must have heard of the penicillin binding protein, the PBP. This is a protein present on the surface of the bacteria where the penicillin goes and binds and then kills the bacteria. What will the bacteria do? It will change the shape of this protein. It has changed the shape of penicillin binding protein to penicillin binding protein 2A, a different type of protein. Now penicillin is roaming about. It is not able to find the penicillin binding protein only. Why? Because bacteria has changed the shape of the target site. Now penicillin cannot bind to the target because it has changed the shape. So these are the 4 very, very important mechanisms by which the bacteria attain or acquire this drug resistance. And the genes which code for the formation of these altered porin channels, these efflux pumps and the target site alteration, these genes get transferred between bacteria through horizontal transfer. And then one bacteria suppose acquired an efflux pump, then now every bacteria around him is now producing efflux pump, everyone's producing efflux pumps, and you're just making the patient drink antibiotic and all the bacteria are just throwing them out and nothing is happening, no treatment is being possible, right? Then we test this. What can we do? We can test this in the laboratory. You must have all heard of antimicrobial susceptibility testing method. So you have 3 options. You can either send conventional methods, old techniques. You simply see what is happening. You expose the bacteria to some antibiotics and you see what is actually happening. So what do you do in conventional methods? Disc diffusion. Basically, agar plate, you've taken the bacteria, you take some antibiotic discs and then you see whether the bacteria is able to come close up to the antibiotic. It is not scared. See, so this is resistant. So I know this particular antibiotic is not going to work on this bacteria because it has grown right up to the edges of the disc. So this is resistance. But if I see a zone of inhibition around the disc, meaning that the bacteria has not been able to grow up till the antibiotic, it has stayed away. This zone, this diameter I will measure, and I have certain standards given to me by CLSI, which is an international body, Central Laboratory Standard Institute. Okay, what does this do? It tells me every year they publish hundreds of pages of documents which tells me about each and every possible microbe or bacteria and its relationship to each and every possible antibiotic, and they give me these zone diameters and they tell me that today if you get a patient whose bacteria is behaving like this with the antibiotic, you tell them whether it is resistant or susceptible. What is important to realize here, every year they have to update these diameters because that is how fast the antimicrobial resistance is happening. If today this year one particular antibiotic was working against one bacteria, to 2026, the same is not going to happen. They are going to become very smart, very, very fast, right? What can we actually do to help it? Then comes the antibiotic stewardship. antibiotic stewardship, which actually is implemented in the hospitals. This takes place inside the hospitals. This is a program which is run. What you can do is, first of all, you have to understand that enhancing infection prevention and control is very, very important inside the hospital. Strict monitoring of hand hygiene, washing your hands, wearing the masks, isolating infectious patients from non-infectious patients becomes extremely, extremely important. Prescribing antibiotics only when they are needed, not prescribing them just so that you can prevent the patient. Sometimes what the doctors do, prophylactically, as a prevention, they give the antibiotics and they say kha hi lo, because you've come to the hospital, some or the infection, other infection you will get. So prophylactically they start antibiotics. That is a very wrong practice, because you don't know which class to prescribe. Randomly you're prescribing high order antibiotics to the patients, right? Then using shortest duration when required. So not if one particular antibiotic needs to be given for 5 days, you prescribe it for 14 days just because you want to be extra cautious. That is not the right method, because you're not being extra cautious. You're simply making the bacteria stronger. So if something needs to be given for 5 days, that information is evidence-based. Thousands of laboratories of data is collected every year, and these zones and doses are decided by standard international bodies. They have worked on many, many patients, and then they have decided it. If you cannot just think that I will give it for extra days to protect my patient, you are not protecting the patient. You are in fact causing more harm to your patient, right? Reassessing the treatment when the culture results are available. So using microbiology labs, using diagnostic laboratories to guide your treatment. Yes, okay, your patient came to you. Most commonly staph aureus causes meningitis in the patients. You start vancomycin. Send the cultures to the lab. The labs will get back to you telling you that this is in fact a gram-negative bacteria. So you have to stop the vancomycin right at that point, right? And then change the therapy according to the report that you have received. Educating the staff, surveillance and supporting interdisciplinary approach. Inside the hospitals, you need to work in cahoots with the laboratories, with the diagnostic labs, along with the regulatory bodies. Now, one important doubt that most of the students keep asking me about antimicrobial stewardship is front end versus back end approach. What is it? It is commonly written in your books. So, front end approach means regulatory restrictions. There are many high end antibiotics such as colistin, which is the last resort antibiotic drug available to mankind. After that, we have nothing left. And most of the patients in the ICUs, they are on colistin. And so, what do we do in hospitals? We restrict their prescription and dispensing. The dispensaries will not release the drug, will not give the drug to the patient or the attendant, the doctor, unless it gets approval by the Antimicrobial Stewardship Committee, the AMSP committee, which is in the hospital, which comprises of senior clinical doctors, microbiologists, pharmacologists, administrative officers. So it is a team of people working together. And then they decide whether you can give this high-end colistin to this patient or not. And then the back-end approach is that this entire team keeps working around in the hospital, keeps taking rounds, keep doing surveillance, collecting data, realizing how many patients we are getting who had MDROs and how did we treat them and what other infection control policies, etc. we need to put in place, right? So that was AMSP, and that is how we can help it. I would conclude this session with this beautiful picture. Suppose you have this melon and you want to cut pieces of this melon. What would you choose? Would you choose a knife or would you choose a drill? If you put a drill onto a melon, of course you will break it, but you will break it apart into pieces and it would be a catastrophe. So you have to choose your weapon wisely. Stronger always does not mean better. It has to be precise, it has to be tailor-made, it has to be made according to patient each time you get a patient, right? So every time using a high order antibiotic, strongest antibiotic does not mean that you're giving the best treatment. So choose your weapon wisely. That's all from my side, guys. Thank you so much for the amazing response you've gotten. It's always a pleasure. I will see you again next time with more such important topics. Until then, take care. Bye-bye.